I intentionally made my talk quite short so that we have plenty of time for questions. In fact, I would really welcome to be interrupted with questions. I know it's a little hard in this format. Maybe nobody will take me up on it. But would be very happy if you want to ask questions in the middle, and then I can pause and repeat them if necessary, and so on. So also want to say everything in the talk today is non-confidential. You can take screenshots, but you can also download the PDF of the slides in case you want to get some of the references that I cite on some of the slides for this. All right, so let's get started. In my lab, which is co-run by Shantanu Singh, we cause problems. No, we create problems. We specifically create computational problems. Specifically, we create computational problems whose solutions will solve very big biomedical problems. So our mission is to facilitate basic biology research, but also drug discovery. And to do so, we're really focused on images as a data source. And we love images as a data source. It's not terribly conventional in biology to use them as anything other than a qualitative snapshot of what's going on in cells. But we love them as a data source because they are inherently single cell resolution. They're incredibly cheap to collect by comparison to most molecular methods. They can be compatible with complex model systems. I'm showing here cells, which are a very complex model system, but you can also work with tissues, with co-cultures of cells, with organoids that are grown in 3D. You can also look at whole organisms. So it's a really compatible with many different systems, and therefore some of the methods that you might develop at one level can be applied very easily to other levels in this multi-scale biology that we live in. They can be multiplexed. In other words, you can measure many different things using images. You can measure protein levels, RNA levels. You can look at of course, localization and spatial relationships between a lot of different things. And then lastly, they're quantitative. And that's really the focus of my talk today, is to hopefully bring your attention to the fact that images are such a nice source of quantitative information about the state of cells. So for context, the data that we're working with comes typically from large-scale imaging experiments, and the basic setup here is that we have multi-well plates. They're usually 384 wells, and the bottom of the plate is clear so that you can image using a microscope through the bottom of the plate and just march around and capture all the different samples. Each individual sample are cells and occasionally small organisms such as C. elegans or zebrafish. They're put into the multi-well plates. Each well gets a different perturbation of some sort, either a genetic or chemical perturbation, and then we snap pictures of how do the cells look as a result, with some kind of stain being added to them. And we then usually carry out some sort of image analysis. It can be classical image processing. Most of the projects I'll talk about today use classical image processing, where we identify different compartments of the cell, like the nucleus and the cytoplasm, and then measure all the features that we can out of those regions. And then we, once we have that profile, the measurements that come out of there, we do data exploration and machine learning, and I'll show all kinds of examples today. So historically, my lab started about 15 years ago, and we are most well known for producing Cell Profiler. Cell Profiler is this image analysis software that's focused on the high throughput world. It's a very modular software, very user-friendly for biologists, and is used by thousands of researchers around the world now, including in the pharma industry, to process what is known as high content screens or image-based screens. The next generation of software coming from the lab is called Piximi, and it is based on really transitioning more to using deep learning, and has all kinds of different capabilities in a web browser format, so very different than the original Cell Profiler. We are happy that these tools are healthy. Piximi is not quite ready for biologist use. It's kind of alpha stage right now, but will be coming out soon. And these tools have stayed healthy, Cell Profiler in particular, and ImageJ as well, thanks to an NIH grant for creating the Center for Open Bioimage Analysis. And so it's unusual for these NIH P41 centers to be software focused. So if anyone has questions about that, I'm happy to provide our experience. And that's run together with Kevin Elisari here at the University of Wisconsin at Madison. Probably our biggest contribution to humanity is creating image.sc. It's an online forum where biologists can come, anyone can come who has a question about image analysis, And if they're stuck on a project, they can ask a question, post their images, and it's such a fervent community, just really lovely people. Oftentimes people will you'll post your question and within 24 hours, somebody's posted a cell profiler pipeline that solves your problem, or an ImageJ macro that can help get you unstuck on a project. So I highly recommend it if you do interact with images and need some help there. So my past work in this area in the field of image analysis has had an impact on patients. And so, I mean, all kinds of really cool projects, and I won't be talking about any of that today, but just wanted to highlight two that have yielded successful clinical trials and therefore patient impact. One was a collaboration with John Crispino from the Northwestern University, where we helped him to develop a pipeline to identify nuclei in images and measure how their DNA content, how polyploid they had become, in order to kind of attack cancer in a different sort of way. This particular type, the idea is to cause the cells to differentiate into a polyploid state, which is going to be less proliferative and less harmful to the patient. And that was very exciting when that showed good clinical results. The other one shown here, the leukemias and lymphomas, we had absolutely nothing to do with. Very cool that these researchers downloaded our software and then used it in a clinical trial in order to guide what individual chemotherapy each patient should receive based on how did their cells respond morphologically to potential candidates that they might be given. So I'm skipping over this all very quickly because what I really want to talk about today is a revolution in how images are being used. So in the examples I showed, and in most of how images have been used quantitatively in biology so far, the idea is that the biologist comes to the image analyst and says, I have a phenotype that I care about. I know what it looks like. I've seen it. I've characterized it. Can you help me quantify it? Either because my paper just went through review and the reviewers are asking me to quantify this thing that I see by eye, or because I'm about to create 100,000 images and I want to be able to quantify what I see by eye in an automated way so that I don't have to do this by hand or by eyeball. And so that field of measuring phenotypes that we already know that we care about, specific individual phenotypes, maybe 1 or 2 or 3 phenotypes in an experiment, that's known as image-based screening, sometimes called high content screening because it's high in information content. That research I worked on for a decade or so and has now transitioned to a former alum of my group. That's Beth Simony. She launched her lab and is carrying on this work, including developing Piximi, as I mentioned before. So what I'm going to talk about today is what the Carpenter Singh lab has been working on for overlapping maybe about the last decade or so, but it has really gained momentum in the past few years. And that's using images as a data source, as a quantitative data source that represents cell state. And we call this image-based profiling. In image-based profiling, we often use the cell painting assay. I'll describe what that is in a second. In this field, the idea is that you capture all the information that you can out of the image and use it as a quantitative description of the image. And there's all kinds of neat things you can do with that. And to get the basic concept, I think it's helpful to think about how diagnoses are made in the world. It's often using image-based information. So many of us lay people who are not MDs can look at a human child and say, I think that kid might have Down syndrome because I noticed certain things about how their facial features are arranged. And a computer can be taught to do the same kind of thing, of course, to better accuracy than a layperson, of course, if it's trained with an MD, with an MD's input. But even more so, if you zoom in and look at the cells or the tissues, sometimes you have to really look hard to find how is this patient different. Some diseases you can't tell by looking at a person's face. You really need to zoom in and look at their cells. We're using this, we're taking this basic concept of the structure of a thing indicates what its function is, and really just scaling it up. We're using cells in a dish because they're much more manipulatable than humans or tissues. And what the idea is, we take perturbations. They can be compounds, in other words, chemicals or drugs. They can be genetic perturbations like CRISPR or overexpression. They can be different patient cell lines. So we prepare the samples however we're going to prepare them. We stain the cells, we image them, and we extract all these features from them. And in doing so, we can measure the similarities and the connections between whatever the perturbations were, whether they're drugs or genes or disease states, and we can measure all these similarities and connections. So if you work on transcriptional profiling, I'm hoping this sounds incredibly familiar to you, because it's really the exact same concept as transcriptional profiling. It's just instead of having single cell mRNA data for every different sample, we now have morphological features for every cell in every sample. And I think the key difference is that we can, a pharma company can easily run this assay on their entire compound library. So 2 or 3 million compounds they can capture this type of data for, and it's quite inexpensive. It's generally on the order of $1 or $2 per well to run these kinds of samples, or $1 or $2 per sample. So this is really key to understanding the whole rest of the talk. I probably should have said that before I went through this slide. So if you've tuned out, you probably should tune back in. This is the whole thing that I'm going to give examples of how this kind of data can be used for the rest of the talk. So I hope it's clear that we perturb cells, we image them, we get their features out, and that serves as a representation of how are these cells experiencing life, what do they look like, and so similar genes should give similar profiles, similar drugs should give similar profiles, and so on. Any questions about this part so far? And feel free to just like raise your hand and shout it out. I can repeat it if you need to. Yeah. The question is, yeah, what cell types? Of course, you can test any cell type you like, and I'll give lots of different examples, but just like transcriptional profiling, there will be certain questions you can only answer in just the right cell type, and other questions that might be more general, and other drugs, for example, that might do the exact same thing in all kinds of different cell types. So that's part of the experimental design. Yeah.
How did you get these features?
How do we get the features? Oh, I'm really glad you asked that. So here I'm showing image analysis. So we often use, almost everything I'll describe uses Cell Profiler, and it's just classical image processing methods like watershed and segmentation and so on to identify the different compartments, extract things like texture, size, shape, intensities, and so on from the cells, and also neighborhood relationships. How are the cells looking? Are they clumped up? Or are they far apart from each other? All those kinds of features are what's measured when we do classical image processing. We also use deep learning to extract directly from the raw pixels of images. And from our lab's experience and from what other groups tell me, you can usually perform tasks more powerfully if you use deep learning for feature extraction, especially if it's supervised or self-supervised in some way. But it's often not dramatically better. It's maybe 10 or 20 or 30% better using deep learning. So we often do a particular project first with the classical features because we know what they mean and because they're very interpretable. Whereas with the deep learning features, it's harder, not impossible, but it's a whole separate project to figure out what features, how are the cells responding to this perturbation when you have deep learning features is a much harder question to answer. All right. Okay, so you might ask, if we are not looking for a particular phenotype, I mean, if you're in the world of image-based screening and you want to measure phospho-S6 levels, or you want to count mitochondrial blobs or something, you know what to stain for. In this case, in image-based profiling, we kind of want to measure all the things that the cell is doing morphologically, but what do we stain for if we're just wanting to measure a kind of generic everything of the cell? And the answer here was mostly dictated by a few criteria, including how cheap is the stain. Number two, how cheap is it? How like general is it? We didn't want super specific stains, and we didn't want to use antibodies because those require more washing steps. So these are just every cell biologist's favorite dyes is basically what ended up in this staining panel. And in the end, it's 6 stains that we image across 5 channels. Sometimes we collect bright field as well, and that adds a little bit of more information than you get with just these stains. And it's staining 8 different components of the cell, and you can see them described here. And again, when we extract signatures from these images, these profiles allow us to look for similarities and matches across samples. All right, so now we get to all the different applications. So at this part, you can just like tune out for one and come back in for another. You won't lose too much if you've got the basic concept. So here is a representation of a drug discovery pipeline that is using cell-based assays. So in this pipeline, you develop some kind of assay to measure some kind of thing. You test a lot of small molecules, a lot of drugs. And then you find some hits. That means compounds that are effective at doing something in the assay, and then you optimize them chemically, the chemical structure of them, in order to produce leads, and then those go into clinical, preclinical studies, and then eventually clinical trials. So we'll begin at the beginning with an example that I think is on the simpler end of the spectrum to understand. Can we identify signatures of disease and then screen drugs to reverse the signature? This project is pretty exciting to me because it's literally has the potential that what we find in this study could impact patient care. So we're working with a collection of rare diseases. This is in collaboration with Mitochondrial at the University of Toronto. His graduate student, Jessica Lacoste, has created a really beautiful set of data using, I'm sorry, I'm gonna skip ahead here. She's overexpressed and imaged 3,500 disease variants together with their reference counterparts, their wild type genes that do not have the disease associated variant. And the idea is here that you first you tag the protein, tag the gene, and make it fluorescent. So in this case, using an antibody, and then you overexpress the same protein, but with the mutation present. And so in this case, it's a mutation that's associated with Noonan syndrome. And if you look at a child with Noonan syndrome, you might not notice anything different. I don't see anything much out of the ordinary. But if I looked at the cells of the patient and looked at the localization of this protein, we might actually notice a difference. That on the left-hand side, we have roughly 3 cells, these black nuclei, these black holes here are the nuclei. So this protein is outside the nucleus and in these kind of vesicle-shaped thingies. And then when this mutation is present, this is just a single cell shown here, the nucleus is here, there's much more protein in the cytoplasm just loose hanging around, and then the structure of these vesicles has changed quite dramatically to be much smaller. So clearly, we may have no clue what this gene normally does, what the gene product normally does. We may have no clue what the impact of this mutation is, but we can see a difference. We don't have to understand the mechanism in order to say, well, now that we've got cells that are in this state and looking unhealthy, we can test drugs and figure out which ones might reverse that phenotype. We can also test genetic perturbations to find potential targets and pathways that might be relevant to understand the disease better, and then eventually to do biochemical screens. And so in this project, Jessica had already found many of these pairs. It's by no means all of the variants are visibly localized differently. It's a very small fraction, but it's a very large number. So even though it's less than 10% of the disease-associated variants have a localization phenotype, it's still a huge number, 200 disease phenotypes that we've discovered so far using this channel, as well as a couple of the more other channels that I've not shown here that are more like the cell painting assay, some just general organelle stains as well. So hundreds of disease phenotypes now. And so each one of these, what we can do now is test existing FDA approved drugs and see whether they cause the unhealthy cells to look more like the healthy state. So very simple concept, but hopefully something that could potentially impact patient care. What's exciting about this, I mean, it is very exciting to have 200 disease phenotypes. However, we're grant funded like everyone else. We don't have grants to run 200 drug screens in an academic environment. It's even for just a few thousand drugs each, it really adds up. So we are working together with Paul Blaney and JT Neal at the Broad, who have been working on pooled optical profiling, where you barcode each of these reagents, each of these wild type and disease variant genes, and you pool them and you test in a given well, you test one compound to see whether any of those diseases will be cured, so to speak, with that drug. So for each drug, we test 200 disease and 200 reference counterparts and try to see whether the drug might be effective for any of them all at once. So that work is still in progress, and it's a good thing too, because there's a consortium at NIH called the IGVF, the Variant to Function Consortium, and we'll be testing 80,000 variants. So again, even if it's less than 10%, it's still going to be a huge number of variants that we could potentially do drug screens for. So that'll be upcoming. Any questions about this kind of a project? Yeah.
I have actually a question about something that came up before. You talk about channels. Are these just different wavelengths or even something else?
The question is whether the channels represent different wavelengths. Yes, this is super, and I should point that out. It's super simple microscopy. Anybody with a microscope that has the kind of normal, has a reasonable filter set can set it up to take images like this. So you can do this experiment, the cell painting assay in small scale. And yes, there's no fancy spectral imaging or anything like that happening here. They're really just the organic dyes that you buy from a commercial vendor and add to the cells, and then use filter cubes to separate them.
This looks like a specific variant for Noonan syndrome. Do you have different variants? Do you have similar profiles, or is it very the same?
The question is whether different variants have different profiles, and yes, actually, if you look at the numbers here, we had 3,500 disease variants, but only 1,200 of their matched counterparts, which means on average we have 3 variants per gene. And so yes, in some cases there's only one variant for a given gene, but for many others we might have 3 to 5 to 10 different variants that are all disease associated in some way. And absolutely, we can look at the similarities and differences among those to see how they group. And we've done an experiment that I think I didn't include in the talk today, but using lung cancer alleles, and the same basic approach using lung cancer alleles, and it's beautiful. You see like all the constitutively active mutants grouped together, all of the not other kind, loss of functions ones will group together as well. So it's a really cool kind of approach to, and very non-gene specific. Normally, if you wanted to do this kind of an experiment, step 1, figure out what the gene does. Step 2, make an assay for the gene, and then step 3, look to see what the mutant does to that assay. But here it's just the same assay across hundreds of genes. Yeah, thanks for raising that point. All right. In another project where we're taking the same approach of can we find a disease phenotype, instead of taking known mutations, we took patient samples, patient skin cells. And this was in a study where we were looking for morphological phenotypes associated with different mental conditions. And here are just, it's real data, but I'm just showing a 2D space, consider it like a UMAP or a PCA or a TISNE kind of plot, where the dots are individual cells. And on the left, we have cells from patients that are healthy. And on the right, we have cells from patients that have bipolar disorder. And as you might be able to see, there's a lack of cells in this region, whereas there's normally an enrichment of cells in the control patients. So there's some morphology, there's some way here the cells are put into space so that similar cells are nearby each other. So there's some morphology, there's some way that cells look, which is very common in controls, but very rare in bipolar patients. And so again, we don't really need to understand what that difference is. I mean, it's fun to look and see what it is, but you can immediately just say, okay, whatever those cells are, can we make them is there any drug that makes them come back again? And so we could potentially test drugs to reverse the unhealthy morphology. Now, in this case, we actually did want to know what the mechanism was. It turned out to be very simple. Like usually an image-based profile is 1,000 features, more than 1,000 features, and it's very, subtle complexity of a lot of different features changing. In this case, we could pretty much narrow it down to a single one that was very dominant, and that is mitochondrial dispersion. So this is just referring to how far away from the nucleus are the mitochondria. And in the case of patients that experience psychosis, which includes bipolar disorder, schizophrenia, schizoaffective disorder, the patients have less of the mitochondria right near the nucleus. I'm sorry, less of it far away from the nucleus. So in a skin cell, you might be asking, why on earth did we do this experiment in a skin cell? And the answer is partly because people do not like giving up live brain cells to you. The only way you can get those are possibly if somebody happens to have cancer and also one of these other conditions and they've had a biopsy, then you might be able to get some, but it's really just impractical. So skin cells are easy to acquire. But what's interesting is if you think about mitochondria extending from the edge of the cell, maybe a skin cell doesn't care too much where its mitochondria is, but a neuron, of course, cares very much more where the mitochondria end up in terms of being able to metabolize and produce energy far away from the nucleus. So we think that's what's going on. Here's the actual data. I've shown you like real data, but this is what the data looks like for the patients with psychosis versus controls. And this project, I should say, started in 2007. So it's as old as my lab, 15 years, but it's because it takes a long time to gather these patient samples. And it's always been a little side pet project. Recently, we've started differentiating those into astrocytes, and so far just have very small numbers, but have confirmed this general trend that we're seeing about the dispersion is also the case for more neuron-like cells growing in a dish. So this is very exciting that we've identified this morphological change. So now let's test drugs to reverse the unhealthy morphology, except again, we don't have funding to do a drug screen. So we thought, well, maybe we don't need to physically do a drug screen, at least not right off the bat. Maybe what we could do is predict compounds that would reverse this morphology, and then we can physically test those in a much smaller experiment. So what we did here is we said, have we ever seen any images, getting back to the topic of the panel discussion just now, have we ever seen any experiments, in any experiments we've ever done before, have we ever seen the mitochondria change their dispersion in any of these past experiments? So reusability of data is turning out to be so valuable in the case of image-based data, because most of it's only been examined for a very small number of particular features, and there's tons more information there. So here's one small data set that we started with mostly FDA-approved drugs that are well characterized. And we looked at mitochondrial dispersion. I'm not showing any of the data that is kind of normal amounts of dispersion. I'm only showing the data points that are very high or very low for mitochondrial dispersion. And the first thing we noticed is there's a lot of nasty cancer drugs there. So we're definitely not giving nasty cancer drugs to patients with a chronic illness. And so there's something else we ought to look at in addition to mitochondrial dispersion. We should also look at whether the cells overall appear to be fairly normal. So that caused us to add this y-axis, which is looking at all the other features that cell painting captures, or the cell painting like assay that we used here, and make sure that those all look pretty normal. And so that led us down to this area of the plot, where in this initial study, we found a couple of things that our collaborators are very excited about, because these particular hormones are known to influence the symptoms that patients have as they fluctuate. And so we're pretty excited that this validates the general idea of looking for compounds with this changes in mitochondrial dispersion. And so from here, we'll be able to look at much larger compound sets as well as genetic perturbation sets to try to figure out potential targets for these disorders. So I've shown two examples of how to identify disease phenotypes using image-based profiling. The first one was looking at localization or morphology changes by tagging the gene and overexpressing it. The second one, we used patient cell lines. And then the third one, I won't go into detail, but you can also knock down genes or just generally overexpress them. And that's an approach taken by a company, originally the University of Utah. They spun out a company called Recursion, and they have 4 candidates entering clinical trials. So this one is definitely further along than our academic lab, but really exciting to validate this basic strategy of identifying disease phenotypes. So any questions on this kind of an approach?
So there are some.
Oh, there's some questions online. Yeah,
There
please.
are some questions online.
Perfect.
I can start with one of them. Fantastic talk. Can cell painting capture morphological changes independent of what one of the 6 stars probe for? For example, some perturbation causing neurotypes. Would that be the same as your bipolar disorder example?
Just that last part.
For example, some perturbation causing neurotypes.
Causing what?
Neurotypes.
Oh, I see. I think I understand the question. So it is absolutely the case. Like you would say, you might think we stain for 6 things. Of course, drugs that are known to impact those 6 things, of course, we're going to find those, but do you get other things? And I would say in general, we found that roughly 70% of FDA approved drugs impact one or more of the channels in the assay. So it's definitely catching a lot more than just the narrow things that you feel like it should be able to capture, which is more a limitation of our understanding of cell biology and how these drugs impact it than anything else. But it does capture a lot of broad things, which I think is what it's asking. I'm not sure I caught the meaning of the rest though.
I think so. And how many patients and cells from the same person needed at least to train the model?
Ah, great. Yeah. So how many cells do you need to get like a good image-based profile? Maybe to bring it to more general point. I would say usually it depends how strong the phenotype is that you're looking for. But generally, I would say you need at least 100 cells, but typically 1,000 is probably the minimum to get to for kind of more subtle phenotypes and the more common case. So personally, it's so cheap to capture these images that typically we have around 5,000 per sample, but it's not really all that. It's only limited by how much time you spend on the microscope capturing the images.
And the last one, if you don't understand the mechanism, how can you be confident that reversing the image-based cells phenotype contributes to reversing disease phenotype? It's a potential limitation.
Yeah, totally fair question. Yeah, you certainly can't be sure. So yeah, I don't want to claim that this is like an immediate you immediately go from a drug that does the right thing to cells in addition to a patient, because there's so many things going on. What does it do to other cell types that you haven't tested? What does it do to other stains that you have not assessed? So yeah, there are still multiple steps from there. However, in a case where you already have FDA approved drugs, that at least reduces a lot of the concerns about toxicity, or depending on the drug, it reduces concerns about toxicity. So if you find, for example, in some cases that I'm aware of, people have found like, oh, hey, it looks like vitamin D might be useful for this disorder. No problem. A pediatrician doesn't mind prescribing like, hey, why don't we boost up your vitamin D levels and see how your symptoms change? Because it's quite innocuous and it's quite unlikely to cause any harm. So in some cases, it'll be immediately actionable to test it in a patient. In other cases, you're going to want to start more preclinical than that and test in other environments.
Thank you.
Yeah, thanks for those questions online. All right, so let's go to a different example. The next one is, can we make better diagnostics using image-based output? And for these, just sort of by chance, these are all going to be label-free experiments. So I talked about cell painting with all these fluorescent dyes. Now we're going to look at some examples where they're totally label-free. And we started out working with some researchers where traditionally they use they use flow cytometry and 4 different antibodies that are fluorescently labeled, and they can distinguish the white blood cell types using just these antibodies. And so we thought, I wonder if there's enough information in just a bright field image of the cell, just using plain light, no markers whatsoever, can we distinguish these cell types based on their morphology alone? And it turns out you can very well, with, as you can see, the 97% here. And furthermore, we could separate B and T cells, which normally the clinicians can't detect the difference by eye very well. We can even do a halfway decent job distinguishing those. So then we got greedy and did the exact same thing in another domain with leukemia. In ALL, these children with this type of cancer, traditionally they'll have 4 to 10 different antibodies, this whole antibody panel to label all the different CD markers on the cells and figure out whether the cells are leukemic or whether there are other normal lymphocytes and other cell types that are supposed to be there. And again, with just this bright field imaging alone, we could classify leukemic cells with pretty high accuracy. So this really has gotten us excited about the potential for, I mean, if you think about it, of course, this means there's great potential in bright field images alone, but it also just makes you realize how much more information is even in the fluorescent images that we've been capturing. Like, you don't want to just measure the amount of fluorescence. Of course, you would want to measure the spatial location of it, because that seems to be a really powerful source of information. The last experiment, I'll go a little bit more into detail. I'll skip that, in fact, because I'll show it here, where we're monitoring the degradation of stored red blood cells. So if you go and donate blood, they put your blood into a bag, they store it at a certain temperature with certain reagents added to it to try to increase the stability. But eventually, after 6 to 8 weeks, depending on the country, they will have to throw it in the trash. And so there's researchers trying to figure out how can we cause the red blood cells to stay more stable and healthy over time, so that we don't need as many donors, and so that we can transport the blood at different temperatures and so on. And so historically, researchers in this area would notice that the cells follow this progression shown here, and they categorize the cells into 7 categories, and then an expert stares at the cells and sorts them and counts up a morphology index that's based on what fraction of the cells are in each of these different categories. Super tedious, and so anybody who's done any machine learning would say, hey, why don't we automate that? It seems like that's something computer vision can do, and it's absolutely the case. It turns out that you can train the computer to replicate an expert. You might be a little disappointed. It's 76% accuracy, but as you see, it's a continuous process and most of the errors are off the diagonal. And we felt even better when we compared, this is the deep learning prediction versus the expert. We then compared the expert versus a different expert, or collection of experts, and they were kind of all over the board too. So 82%. Here, I think we can claim that the computational method is roughly the same as an expert. But we also realized maybe we don't want to match the expert in their ability to categorize these 7 categories. It is a continuous process. So perhaps we can do something a little smarter here. And what we did is we trained a deep learning model on the number of days to predict the number of days of storage of blood. Now, a classifier that can do that is completely useless for anything in real life, because unless you have like a pile of bags of blood that you don't know how old they are, I mean, they should have labels. You should already know how old they are, right? So you don't care about the classifier, you care about the features that the model has learned to extract from the images, because those correspond to some biological meaning. You know that the time in storage corresponds with some biological changes of degradation that are bad generally. And so this should be able to extract those kinds of features. So extracting those features, we could then create this self-learned morphology index score, this like linear model of how the cells age over time. And we found that score, that self-learned morphology index, correlated better with the y-axis is showing here the sort of real performance of the blood. It's an expensive assay to do, or a difficult assay to do, to get the hemolytic score about how the cells respond to use. And it corresponds much better than the original expert manually defined morphology index. So we're really excited here. There's a particular toolbox that makes this kind of experiment more easy to carry out, but really excited in general. I think the general principle here is it's wonderful if you can find something biological to train on that is not humans in the cases, so that we're not relying on the biologist's perception of what might matter. All right, next, any questions on those series of label-free diagnostics? All right. Yeah.
Did you attempt to visualize the features?
Oh, did we attempt to visualize the features? No, we didn't. And in this case, it probably wouldn't be so interesting to do so, because you know the clinicians already knew that you can kind of, you can see some obvious changes over time. So of course it should recover that. So anything that it didn't recover would probably be pretty subtle and probably difficult to look at. But in general, I love the concept, and in other scenarios, it would definitely be something we would try, but we didn't do it here. Sorry, I'm not sure I heard. Yeah. So the question is whether we could interpret the deep learning features to understand the differential between what the deep learning could get and what the humans saw. And no, we didn't attempt it, but it would likely be pretty subtle and a bit of a challenge.
All right. Very basic question. What's bright field?
Oh, what is bright field? Bright field is just when you shine light on cells and snap their picture. And just the example, these are examples shown here. So they're not shown as, they are black and white images, but they're not like the fluorescence images that have the black background and then some color on top. Instead, it's just these, and here's a few more examples here. It's just like plain light images. So very cheap. You don't have to stain the cells with anything. You're just, how do the cell's material, the absorbance of the cell itself, how does that respond to plain light? So it's cool. In these cases it doesn't matter so much, but it's cool because bright field imaging could potentially be compatible with, let's say, a really cheap camera that connects to a really cheap microscope that connects to a cell phone camera and has some built-in software that can do some kind of diagnostic in a resource poor area. That's where you would really care about an assay that is using bright field only. Apparently all the people on this side of the room have questions. Everyone else. All right. I'm sorry. Okay, so the next example is, can we identify gene and allele functions by grouping similar genes and alleles to each other? So here the idea is you profile a query gene. Here's a gene I care about. When I overexpress it or knock it down, and add it to cells, the cells respond morphologically in some way. And whatever that pattern is, I want to go and look, have I ever seen this pattern before? So it's all about matching your query to a set of known samples. And so the first experiment we did in this vein was about 5 years ago, where we overexpressed each gene, did the cell painting, did the image analysis, and then clustered the profiles. And it was a beautiful sight to see all the different biological pathways in the cell in this one really simple and cheap experiment. So we saw proteins that bind each other, were right near each other and showed very similar phenotypes often. We saw pathway members nearby each other. We saw negative and positive regulators of a pathway giving opposite morphological profiles, which was very cool. We saw mutations in proteins. If it was a constitutively active one, it looked similar to the wild type, but definitely different enough to be nearby, and so on. So really exciting to see this, and made us want to scale up to the whole genome, which just this year we've made data from 13,000 genes, which will be released in November, and I'll describe that more in a bit. So that's pretty exciting. What can we do with it? Well, first of all, we can look at alleles. And as mentioned before, with the question about alleles, we've done a whole project on lung cancer that's not shown here. The other thing we can do, aside from figuring out the function of genes that we didn't already know, is we can potentially identify chemical regulators of genes. And so here we put the gene profile in, and we hope to find chemical profiles that match. And in this project, we again, very inexpensive computational virtual screen where the input is the profile from a gene that's been overexpressed, and the output is any matches that we can find in a big database of compounds that have been added to cells and where their morphology has been measured. And in this case, we have 30,000 compounds. Roughly half of them have do something to cells. So that's the pool that we're searching against. And first we validated it against known gene compound pairs, and that's shown here. If we rank order the list of 30,000 compounds, half of them are out of the running right off the bat, but then for the rest, rank order each gene's top matching compounds, and is the correct answer, is a correct compound in that list. And so the analysis is shown here, where 32% of the genes that we tested had good compound hits at the top of the list. From a pharma company perspective, or from any of us' perspective, this is quite a nice ratio, because if you're telling me that instead of physically testing 1 million compounds, I can instead just look up and query in the database to get a list of, let's say, 100 candidate hits, and then just go test the 100 instead of the 1 million. This is a huge money saver. Even if 60% of the time it fails, the 32% of the time it works really makes this pay off. So in a pharma context, their assays typically cost somewhere between $1 to $5 a well. So if you're saving screening 2 million compounds, it adds up. It's a lot of money. So that's all very nice to use existing known gene compound pairs to validate the method. But we wanted to prospectively test it. It's easy enough to predict stuff we already know. Let's make sure we can actually discover something new. So what we did is we found, we made these lists. So we made these lists for every gene in this experiment. Here's the list of compounds that we predict are regulators. We went around and talked to all of our friends at the Broad and beyond and said, hey, you study this gene. Would you be interested in some compounds that potentially regulate the pathway? And we found 7 people who are willing to help, which I think is already an interesting social experiment. I think we asked 9 people and 7 were willing to do these experiments, which was an awesome hit rate for people being interested collaborators. And out of those 7, we found 3, again, this is like roughly 30 to 40% of the time, it worked. They found, in fact, there were two of the 7 cases, 5 of the cases total, they had some promising results. 2 of them, the chemistry was bad. I'll spare you the details. But 3 of them convincingly worked, where we found inhibitors of particular pathways that were interesting. The one that's most interesting is shown at the bottom. YAP1, we found 3 really good pathway regulators that are shown in blue in the plot. And the assay here, where we were testing them, was to see whether these compounds could kill YAP-dependent sarcoma cells. Sarcoma is a really nasty kind of cancer that has a poor prognosis. And being able to kill these cells specifically without killing the rest of the human cells would be a really big strength. And so we found not just that those 3 original hits, but then some chemical variants of those hits. We found some even better ones that really do a great job of killing these sarcoma cells. And we're hoping to follow this up with the National Cancer Institute program to further develop these compounds for sarcoma. Any questions about this virtual screening where we put a gene query in and we get compounds out? Yeah.
So how do you avoid the off-target effect? Is there selective inhibitors?
How do we avoid the off-target effect? And what was the last bit?
And are those selective inhibitors?
And are they selective inhibitors? Okay, great. When you treat cells with a compound, he's referring to off-target effects, which means even with FDA-approved drugs, much less regular chemicals that people that have synthesized, they're often very unoptimized. In other words, you add the chemical to the cells, they might attach to multiple proteins and then have multiple effects, some of which you would call on target because they're what you wanted, and some of which are off target, which you wish did not occur. Some drugs are on the market that hit multiple targets, and that actually turns out to be a good thing because it has the right effect in the cells. But in this experiment, our goal is to find something that looks as close as possible to YAP1 overexpression. And so we have no idea what it might be. It might be hitting YAP1 specifically, but it might be doing something upstream or downstream. All we know is that it mimics the morphology. And so it's absolutely the case that this is not going to yield super specific inhibitors of just that one protein. We do have some confidence that it probably doesn't hit a lot of different things, because if it did, it would yield a fuzzier profile that would not match YAP1 so well. So in that sense, it's like maybe a bit more specific than you might otherwise think, but it's by no means a guarantee. So you would still, for these kinds of compounds, you still want to check and see, figure out what exactly is the direct target, and are there any off targets that would be of concern. The one thing we could do, the Broad has a really amazing platform where you give them a drug and they'll test viability against around 1,000 cell lines. And we found in this case, they don't kill very many cell lines. It's really, it's not all sarcomas. And there are a few other cancer types that it does work against, but it's relatively specific to those cancer types. So no idea what they bind, but at least it's not a widespread killer.
Yeah, very nice presentation. Very nice presentation. I just want to have two quick questions. One is, have you compared your imaging kind of like reversal signature with the connectivity map, the gene expression signature? Because I know A549, they do have a profile. Like you see kind of like the similar drugs is really kind of like go back to the top and then have similarity. And the other thing is, because in the original connectivity map, they are doing some sort of like a ranking base to do this kind of matching. And then how do you do this for your feature? Because they are kind of like different measurements and different cell type. Do you have some kind of like insight that you can share?
Yeah, I love this whole line of questioning. So, so mRNA profiles and image-based profiles. We've only directly compared the L1000 mRNA profiles, which are measuring only 1,000 mRNAs, not a full gene chip, not RNA-seq, although there's some studies that indicate that they're equivalent to RNA-seq bulk profiles. But in any event, the L1000 mRNA is what we've compared directly to. And the feature space is very different between measuring distinct mRNAs versus these image-based features, many of which are quite redundant. So we do need to do some sorts of pre-processing to try to make them more resemble each other. The way we measure similarity with images is using usually Pearson correlation, whereas my understanding is in CMAP, they pick something like, forgive me, I don't remember the details, but it's something like, what are the top 10 and the bottom 10 genes or something like this that go up and down. So it's kind of limiting, it's kind of making it a binary decision about a certain proportion of the genes. So we do that a bit differently as well. And so actually this next project, even though it's not, will address your question of have we ever compared the two, we have across a number of different sorts of tasks. And I'll show one here, which is, can we assess the mechanism of potential drugs based on their profiles? So in this type of application, you have a query compound, you don't know how it works, you know it's useful for something because you tested it in some other assay in pharma industry or something else. Like this drug is potentially useful, but I don't know exactly what it hits. And so you check and see, does it match anything that I've ever seen before? So this is a pretty commonly been done in images stretching back more than a decade now. You'll find papers where people have done this successfully using image-based data. And usually it's matching a compound to another compound, an already known compound, to figure out its mechanism. So I won't cover the whole history of the field, but what we did is we had both mRNA data and cell painting data available for the same set of compounds. So we thought, here's a task where we can compare these two methodologies against each other. So this is a dose response curve, which shows that, of course, as the dose of the compound goes up, we have a better ability to see that compounds that have the same mechanism do actually look like each other. So it goes up to about 30% when you have both data types available and you can use them both and combine them both. But as you can see, cell painting, image-based data very much holds its own against mRNA. If you asked me to predict this outcome 15 years ago, I would have said, I'm absolutely sure that images are not going to be as powerful as mRNA, but at least they're cheap. That would have been my statement. And so we were really surprised to see not only that it's holding its own, it actually seems to have more information content. And this just gives you a sense of which mechanisms are captured better by one or the other. And you can see most of them are roughly along the diagonal that, sorry, I should say most of them are undetectable by either. Roughly 70% of the categories you can't see very well the right mechanism with either one of them. But then among the rest, many of them could be detected by either one. So to some degree, these data types overlap, but to some degree, there's some distinctive information there as well. And there's all kinds of really cool analysis that Greg Way did in the lab, comparing and contrasting the two data types and their strengths and weaknesses. All right, so I have one more application, which is, can we predict the outcome of a complex assay by correlating its readout to cell painting profiles? So this one's a little more machine learning intensive, and I'll show you what I'm talking about here. First, you need to understand something which I already find mind-blowing that it works. It is already the case that if you take the structures of compounds, you feed them into a deep learning network together with assay outputs, you can train this network to learn to predict which structures are likely to produce positive effects in which assays. That is very cool. It of course requires a lot of data for training so that you start to learn what parts of the chemical structures are yielding which outputs, but this is very cool. To me, it feels completely unfair to expect chemical structures to reveal much of anything because there's just so much going on between the chemical and the output in the cell. So I feel like we should give it some more information. So we thought we have a set of 17,000 molecules where we have both the image data and the mRNA data. And so here's another example where we can compare and contrast the two. And what we found is, okay, let's give the network not just the chemical structure, but also the images that are produced when you treat cells with that compound, as well as the mRNA profiles that are produced when you treat cells with the compound. So when we do this, it definitely does not work. I should say, predicting assay outputs from chemical structures, again, is kind of a single-digit % of the time it actually works. But it turns out, adding the images or having each modality alone, it turns out the morphology data, the image-based data, is very effective at predicting, and in fact, better than the mRNA data. And we did all kinds of analysis. You can check the bioRxiv preprint about this. In the end, if you have all three data types, you can end up predicting roughly 15% of the assays. 50 is around 15% in this experiment. So there's a decent proportion of biology that can be predicted pretty well. Again, speaking of the percentages here, 15% might sound like a very pathetically low percentage, but each of those represents an assay that costs $5 to $10 million that can be avoided because it turns out the information in the raw images can tell you whether a certain compound is likely to produce the right outcome in an assay that you might be interested in. So those are all the applications. I'll just leave you with the fact that we're going to be putting a lot of data out in the public domain as of November 2022 as part of this Jump Cell Painting Consortium that we've been leading together with a group of pharma companies and another nonprofit. And it will include lots of small molecules as well as overexpressed genes and CRISPR knockdowns, all in an arrayed format. And there's a website here where you can sign up to a newsletter and be able to be kept up to date as this data comes out. I'll skip that and just leave you with, if you're interested in this field, there's a review article here. There's conferences that meet specifically about using image data. I had to skip several projects, including this disease-associated allele project. You can see the citations there. You can grab this link to look up the slides if you want to look up any of these articles. And with that, I'm happy to answer any remaining questions that you might have. Hopefully we got most of them out of the way. Thank you so much.
Hi, thank you for your talk. I had like a question about the methodology. So
Sure.
I guess you're in image space, you're right adjacent to this like rich field of computer vision. And I wonder how easy is it, do you find it a fruitful direction to draw insights from that community and apply them to the cell space? Is it fruitful, and how generally do you find the mapping to be, how complicated is it?
Oh, wow. Yeah, absolutely. Like not just fruitful, but 100% necessary. So I would say there's very little innovation that's been done on the biology side of this equation. For the most part, the entire field of image-based profiling mostly takes methods that have proven effective in general computer vision tasks, which is much better funded and staffed. And we just adopt them and maybe adapt them to some degree and apply them. So really excited, especially about developments in contrastive learning, seem to be most promising for our field. But yeah, for the most part, we are on the receiving end.
Have you tried this on TMA slides or like H&E images? Extracting some of these features?
Yeah, we've I think our lab, I think it's fair to say our lab has mostly stayed away from pathology because there's a whole universe of people working exclusively on digital pathology. But I will say many of the concepts are exactly the same. Again, that's another field that we can borrow from because many of the tasks are the same, especially can I distinguish disease versus healthy is something they do all day long and work on methods for.
Thank you. Very good talk.
Thanks, Sam. Beautiful talk. Question for you. Thinking about like the drug screens, right? There's been large scale differences in batch effects noticed, particularly in the genomics-based side of that. Have you noticed anything in cellular morphology or sort of? Yeah,
for sure we have batch effects, and that's the one slide I skipped. So if you want to, download the slides, you can see the latest. Where again, here we've taken methods from single cell RNA-seq. Again, a much bigger field and a lot more work going on there. So we took Fabian Theis's recent evaluation of all the single cell batch correction methods and just directly applied it and picked the best one and moved on with our lives. So we really are appreciative of folks working in adjacent fields here.
Thank you very much for a very inspiring talk. Maybe a main question. Is there any way to enrich this microscopy with doing in any way 3D picture? Like, can you do different layers and then add this to your data sets, both for screening and for?
Yeah, for sure. There's everything I described could equally well be applied to time-lapse images or 3D or both. Usually the limitation there is just the physical limitation of how long it takes to capture the images or create the samples if you're talking about 3D organizers. So we personally haven't done experiments that use those two data types in this kind of a profiling way. But it could absolutely be done, and there's I've seen a few inklings of people starting to do that work. It really just, I think we're in the low-hanging fruit stage, and for us, there's been plenty of low-hanging fruit with the 2D static images, but I think that'll be a direction that the field is moving towards.
And maybe a related question, since you mentioned the timing of the sample on the microscope maybe is important. Is it also degrading the sample in any way? Is it a concern at all for generating your data?
Sorry, is what degrading the sample?
Like how much time the sample spends under the microscope?
Oh, no, it doesn't. Actually, for the cell painting assay, the samples are fixed, so it's very, very much not a concern.
Oh, I see.
Yeah. Thanks
Thank you.
for clarifying.